are going to our experiment number 1, that is to prepare a temporary stained mount of onion peel and human cheek cell. Now, what is the difference between the human cheek cell and onion peel? Human, it is an animal, social animal. So, by studying human cheek cell, we will be studying about animal cell. And by studying onion peel, we will be studying about plant cell. Now, as this is the cell of prokaryotic organisms. Now, what are prokaryotes? Prokaryotes are the primitive celled organisms. What is the meaning of primitive? Primitive means when the organelles are not well defined or the organelles do not have a well defined boundary around them, then the organisms, they are called as prokaryotes, and the cell is called as prokaryotic cell. Now, if we talk about a eukaryotic cell has a well defined nucleus which is present in the center of the cell and which controls all the activities of a cell. This nucleus, it has a nuclear membrane which is shown here. This is the nuclear membrane, and inside it has nucleolus. Now, after the nucleus, what is present in the cell? Mitochondria. It is the powerhouse of the cell. It also has well defined boundary, means the membrane are there, outer membrane and inner membrane. Now this outer membrane, it is continuous, whereas the inner membrane, it is a folded structure. Mitochondria, which is the cell organelle which is found in the eukaryotic cell, and it has well defined boundary. That's why it is present in the cells of eukaryotic organisms. Now the next organelle, it is lysosome. Lysosome, it is also known as suicidal bags. Then these lysosomes, they are also have well defined organelle. It is also a well defined organelle, means well layered structure it is. Then after lysosome, Golgi apparatus, Golgi bodies जो क्या करते हैं, which helps in storing most of the materials which are secreted by the endoplasmic reticulum or the ribosomes. So they store the useful things, Golgi apparatus, and here what we are able to see that these are stacked over one another. So they form a layer like structure, and these were first discovered by Golgi, and that's why they are called as Golgi apparatus. Now the next organelle which is present in a cell, it is endoplasmic reticulum. Endoplasmic reticulum, it provides the framework to the cell, and it is present just near the nucleus. So here it is nucleus, and attached with the nucleus, these are the endoplasmic structures which are present. These endoplasmic structures, they are of two kinds, as we have studied in our theory, that if they are without ribosomes, means they are like this, then these endoplasmic reticulums, they are known as smooth endoplasmic reticulums. And if they have this dot like structure present on their boundary, then this type of endoplasmic reticulum, it is called as a rough endoplasmic reticulum. Now, why these endoplasmic reticulums are called as rough? Because they have these ribosomes. Ribosomes are the dot-like structures which are present on the walls of endoplasmic reticulum. Now, the next organelle, if we talk about, that is mitochondria, Golgi bodies, endoplasmic reticulum, then vacuoles are there, nucleus is there. So, these are the various organelles present in the cell. And when we compare animal cell and plant cell, means human cell and plant cell, what is the basic difference? That the plant cell, they have cell wall around them. So in a plant cell, what is present? This is a plant cell, and it will have an extra covering that is called as cell wall around itself. Whereas animal cell does not have any cell wall, only cell membrane is there. So what we can say, that cell membrane, it is present in both animal cell also and plant cell also. But these plant cells, they have an extra covering that is known as cell wall. So this is present only in plant cells. Now the second difference is, here we are able to see in the diagram also, that this is vacuole, which is very small in human cell or animal cell, whereas in plant cell this vacuole is very large in size. So what is the function of vacuole? It stores the waste products of a cell, and as human body has excretory organ to remove the waste from the body, so the human beings or the animal cells, they have very small structure or size of the vacuole, whereas in plant cells the size of the vacuole is very big because they do not have any special excretory organ. Now, cell wall, as we have just talked, cell wall, it is present only in the plant cell. So it is the distinctive feature of the plant cell. It will be present only in the plant cell, and we can easily identify whenever we observe a cell under a microscope, and if if it has a cell wall, then we can say that it is the cell of a plant. Now, plant cell has a well-defined cell wall, membrane is there, nucleus is there, and cytoplasm is there. So, this is a plant cell. So, this is the cell wall. Inside cell wall, it has one more layer, that is cell membrane. Then nucleus is there, and it is filled with the cytoplasm. So, this about the plant cell. Now, and one more difference is there, as we know that plants are able to do photosynthesis because they have green colored pigment, and that is known as chlorophyll, and because of this chlorophyll they are able to trap the sunlight or the solar energy. But now the question comes that where this chlorophyll is present. We have studied that it is present in the green part or in the leaves and the green stem, but this chlorophyll, which is a pigment like structure, it is present in a special organelle, and the name of that organelle is known as chloroplast. So chloroplast, it contains chlorophyll, and this chlorophyll helps in photosynthesis. How it helps in photosynthesis? It traps the solar energy and converts it into chemical energy. Now, this is all about the theory portion, that what is the basic difference between a plant cell and animal cell. Now, our experiment is to prepare a stained temporary mount of an onion peel. This is the first experiment and A part. So, A part is to prepare a temporary stained mount. Now, we will understand the meaning of these three terms. Stained means colored or coloring. Temporary means because the slide we will prepare in our lab, so it is not a permanent slide, so we will say it is temporary. And mount means slide preparation. So when the slide is temporarily prepared in the lab by using the coloring materials, then this is known as stained temporary mount, and we will prepare the slide of onion peel. So what are the materials required for preparing this slide? Materials required are, this is slide. It is made of glass and it is known as slide. You can easily identify it. It is a rectangular shaped transparent glass object, and the name of it is slide. Now the next material required, it is glycerine. Now what is the function of glycerine? As it is shown here, glycerine, it will not allow the air to enter into the under the cover slip when we will doing this experiment. So it is glycerine. Now the next material required is, these are the pieces of onion we have taken, and we will peel it off. We will remove the peel, and then there is a transparent membrane present on the inner side of the onion. So we will remove that peel. So it is onion. Then next is cover slip. So this is this picture. It is of cover slip. COVER, cover slip. So what is the function of this cover slip? This, when we prepare a temporary mount on a slide, Whenever we keep the object in the center, then we use this cover slip for covering this slide so that dust particles may not enter. So it is called as cover slip. Now the next, it is distilled water, which we will be using while preparing the temporary slide. So it is distilled water. These are the materials required which we are talking about and which we will be studying in our this experiment. Now next, it is a watch glass. It is a watch glass and it is transparent. Its function, or what is its use? It is used to hold the water in it. Next material is safranin solution. Yes, you have to learn its name. Safranin, it is a coloring agent, because in the name of the practical we have talked that stained temporary mount. So we can say it is a stain or the coloring agent, and the name of it is safranin. It is pinkish color solution and it is used to put the to color the onion peel, because onion peel, it is transparent, it is whitish in color, and when we will observe under microscope, we will not be able to see its various organelles unless it is colored. So for coloring this onion peel, we will put the drop of stain, and the name of the stain is safranin. So you have to remember this thing, that whenever we are preparing temporary stained mount of a plant cell, then we will be using the staining material, and the name of that material is safranin. Next, it is forceps, which is used to tear off the peel from the onion. Next, it is needle, which is used to pick and tear the membrane. Next, it is blotting paper. What is the function of this blotting paper? Its function is to absorb extra water. So it is blotting paper. When we will prepare the slide, when we will put the cover slip on the slide, then whatever the extra amount of water comes out, it is used to absorb that extra water. Then after blotting paper, this is the compound microscope. This microscope is used to see the image of the onion peel. We will be observing it under the compound microscope, and then we will be able to see the cells of the onion peel. So this is all about, this is a dropper. This is also the material required while preparing the slide. Now what is the procedure? How we will prepare the slide? Procedure is, first we will cut the onion, we will peel it off. हम इसका जो छिलका है इसे रिमूव कर देंगे. Now after peeling off the onion, then after holding it from the inner side, we are able to see a thin transparent membrane, and that thin transparent membrane is removed from the peel. So this forceps, it is used to remove the inner membrane of that peel. Now we will transfer that inner membrane into the water, because it is very thin and it can dry off in the air. So we will take the watch glass, and water is there, distilled water, and we will transfer that membrane in the distilled water so that it may not get dry off, because we have to observe it under microscope, and it should be straight, it should be wet. Otherwise, if it will get dried, then we will not be able to see the parts of the cell membrane or onion peel. Now next, it is the coloring material, that is safranin. So what is done? Next, a separate watch glass is taken and few drops of safranin are added into it, and now we will transfer that membrane from और अनियन पील फ्रॉम दैट वॉच ग्लास विच वॉज हैविंग वाटर, जिसमें पानी था, उससे अब हम वी विल ट्रांसफर इट इनटू द अनदर वॉच ग्लास विच इज़ हैविंग द स्टेनिंग मटेरियल. Now we will take the brush and we will transfer it again into the water. Why? What we are doing? We are transferring again this membrane, this onion peel from this watch glass which is having coloring material to the next watch glass which is having वाटर. क्यों करते हैं? ताकि जो एक्सेस स्टेन है दैट इज़ रिमूव्ड. सो फर्स्ट हमने उसे वाटर में रखा था ताकि वो ड्राई ना हो, फिर हमने उसे स्टेन में रखा ताकि वो कलर ले ले, एंड नाउ अगेन वी विल ट्रांसफ़र इट टू अ वॉच ग्लास विच हैज़ डिस्टिल्ड वाटर सो दैट एक्सेस अमाउंट ऑफ स्टेन इट इज़ वॉश्ड ऑफ. नाउ with the help of the brush, Now we have transferred it into the another watch glass of water. Now we will transfer it on a slide. अब हमने जो है मेम्ब्रेन को, इस जो अनियन पील है उसकी जो इनर मेम्ब्रेन है उसे हमने ट्रांसफ़र कर लिया है कहाँ पर, स्लाइड के ऊपर. एंड नाउ वी विल पुट अ ड्रॉप ऑफ ग्लिसरीन. वाई ग्लिसरीन इज केप्ट? बिकॉज वाटर may get evaporated and the slide may get dry off. So what is kept? Glycerine is kept so that this membrane does not get dry off. क्योंकि पानी से या स्टेन से हमने इसे निकाल दिया है. Now we have kept it on a slide. So we will put a drop of glycerine so that it may not get dry off. Now glycerine is there. We have transferred the onion peel inner membrane. And now what we will do, we will keep the cover slip as it is shown here. This is the cover slip which is kept. This is a slide. Then the material or the onion peel membrane is taken, which is colored. It is covered with the cover slip. And now whatever the extra water has come out from this cover slip, it is wiped off with the blotting paper. So this is the blotting paper and it is folded here. And why it is used here? It is used to absorb the extra water or the glycerine which is coming out of this cover slip. Now we will keep this slide under a microscope and we will observe it under the microscope. So this is all about the procedure. Now observation, what we will observe under microscope. Under microscope, because it is onion peel which is a plant cell, so we will be able to see the cell wall, nucleus and cytoplasm is there. Because cell wall is present in the plant cell, then nucleus is there and cytoplasm is there. So these are the parts which are visible in a plant cell. First is cell wall, which is the outer membrane. This is the cell wall. Inside it is cell membrane. And then what is there? These are the nucleus which we observe in a plant cell when the cell is observed under microscope. So these are the different parts which we have talked about: cell wall, nucleus and cytoplasm. Now what is the conclusion? We have prepared a temporary stained mount of onion peel. We have observed it under the microscope, and now what we have observed, that is the conclusion. So what we have observed, that it has cell wall, it has cell membrane and cytoplasm is filled inside it and nucleus is there. Now what precautions we should take while preparing the slide? So first precaution is use a brush to transfer peel from one apparatus to another. As we took a watch glass, it was containing water and membrane was also there, onion peel membrane was also there. So while transferring it to the another watch glass, what we have to use? We have to use a brush so that we can easily transfer the onion peel membrane to the next apparatus. This is the first precaution. Then second is staining of peel should neither be dark nor too light. So that's why what we have done, when we transferred into the watch glass containing stain, after that we washed it so that we can easily see the various parts or various organelles of the cell. So the staining should not be very dark or it should not be very light. Then third precaution is extra glycerine stain should be removed by using blotting paper. So what is the function of blotting paper? That whatever the extra material has come out, it should be wiped off with the help of blotting paper so that the slide look neat and clean. So these are the various precautions, and this is about the experiment number 1 part A. Now the next experiment is, that is experiment number 1 but part B, in which we will study about the animal cell, and for studying animal cell we will be using human cheek cell. Human cheek cells means whatever the cells are present inside the oral cavity or the mouth cavity, वहाँ से we will remove few cells and we will observe under the microscope. So what are the materials required? One is toothpick. This is the toothpick which is used to scrape the inner portion of the mouth. जो हमारे cheek हैं उनके अंदर side पर हम इस टूथपिक से हल्का स्लाइटली वी विल रब इट इनसाइड आवर ओरल कैविटी एंड सम ऑफ द सेल्स विल come out with this toothpick. So next material, it is watch, this is cover slip, and we have talked about it in our experiment number 1A also. Cover slip, it is made of glass and it is transparent. Now next it is distilled water. This is brush which will help in transferring the material. Next is, it is a pen which we will use for transferring the things or for scrapping. And next it is compound microscope. So this is all about the list of materials required or the apparatus required. Now what is the procedure for preparing the temporary mount of human cheek cell? So first we will do, we will take a slide. We will put a drop of this distilled water on the slide. Now what is being done? A toothpick is taken and the inner side of the mouth. This is the inner side, oral cavity, inner side jo cheek ka hai, that is scraped off with this toothpick. Now some of the material will come out with this toothpick and it is transferred directly to the slide. So here with the toothpick, the material is transferred to the slide. Now we will put the staining material, and here which staining material we will use? Methylene blue. So methylene blue is taken for staining this slide. Now here, as you are able to see, because we have used the stain, that is methylene blue. Now what is the question? See, for staining the plant cell we used saffron in solution, but for staining animal cell we are using methylene blue. So methylene blue is used, and because of that the solution is appearing bluish in color, and now we are putting this cover slip with the help of this needle. Now we have to take utmost care that there should be no air bubbles while we are transferring this कवर स्लिप. जब हम कवर स्लिप लगाते हैं हमें बहुत ध्यान रखना होता है, और इसके लिए हम निडल की हेल्प लेते हैं, ब्रश की हेल्प लेते हैं, ताकि कोई भी एयर बबल्स इनसाइड ना रहें. अदरवाइज व्हेन वी विल सी अंडर द माइक्रोस्कोप लॉट ऑफ एयर बबल्स विल बी विजिबल इन द स्लाइड. So now the slide is this covered with the cover slip, and extra water or extra glycerine and extra staining material, it is wiped off with this blotting paper. And after that this slide is kept under microscope and it is observed under microscope. And when we observe it under the microscope, what we see, that a human cell has cell membrane and nucleus. Now you can easily differentiate that there were 3 labelings, and the first labeling, it was of cell wall because that was onion peel cell, but it is animal cell, so it has 2 labelings. One is cell membrane and nucleus. So this is somewhat the structure, irregular shaped structure of human cell which has nucleus inside it, and it is bounded by cell membrane, and there are no cell walls present in the human cell. Now conclusion. We have reached to the conclusion that a human cell, which is representing the animal cell, it is bounded by a layered membrane, and that is called as cell membrane, and inside it has nucleus which controls the various activities of the cell. Now precautions. What precautions we should take while performing this experiment? First, ensure toothpick used to scrape the cheek is clean so as not to cause infection to the cheek. Whatever toothpick we are taking, it should be free from germs, it should be neat and clean, because we are putting it inside our oral cavity, and if it is contaminated with germs, so we may get suffered from the disease. So we have to take it under consideration that the toothpick should be very neat and clean. Now second is, extra stain should be removed by using blotting paper. What is the function and why we use a blotting paper? So that extra stain is removed, extra water is absorbed, and slide is neat and clean. So this is all about the 2 experiments which we have done today. First is to prepare a temporary stained mount of human cheek cell, and to prepare a temporary stained mount of onion peel.